try to find one if you can. So I will give Dr. Fahrenfeld a quick introduction, and then I'll hand things over to her. Okay, so thank you guys for coming to the second WISE seminar of the semester. Again, don't forget to sign in. It helps us track participation. And also, if you are getting any type of extra credit, right, those professors will get that attendance sheet. All right. So Dr. Fahrenfeld is a professor of civil and environmental engineering at Rutgers. She earned her bachelor's degree in environmental engineering from Johns Hopkins, master's degree in environmental engineering and science from Clemson, and Ph.D. in civil engineering from Virginia Tech. Her lab at Rutgers focuses on applied environmental organic chemistry and microbiology to promote water quality with applications in natural, agricultural, and engineered systems. Her work has been funded by various agencies, including the NSF, CDC, and NOAA. And she has been recognized with awards from various groups, including the New Jersey American Water Works Association and the American Academy of Environmental Engineers and Scientists. In addition, she was recently awarded with an Outstanding Faculty of the Year award from Rutgers. And on top of her research responsibilities, she also acts as the undergraduate program director for environmental engineering at Rutgers. Today, she will discuss just a snapshot of some of her research on microplastics, as well as some of her professional journey. So on behalf, of course, of the WISE program and all of our students, we thank you for traveling here. It's always beneficial, I think, for our students to be able to have that interaction in person with our speakers. So thank you for that. And then one more note for our students. If you do have a question you'd like to ask during the seminar, just raise your hand, but we will also have a Q&A immediately following the seminar. There is also a QR code, a couple papers in the back where there's a QR code. If you didn't scan it on your way in, please scan it on your way out. Okay, so answering some of those questions helps us continue this program and maintain our funding. So if with the most of you, right, that we can get to do that is great. Okay, Dr. Fahrenfeld.
Well, thanks for having me today. My lab does about half microbial research and half microplastics research. So I'll tell you about the microplastics today. Have folks heard about microplastics in the news? How concerned are folks? Very concerned, not concerned, somewhere in between? Concerned. Okay. I go back and forth between like concerned and agnostic, and be happy to chat about that. So we've known that tiny pieces of plastic like microlitter has been in the water since the 1970s. And a question I get often is like, why are we talking about this now instead of back, when folks first noticed that? So I think there's a few things that came to head recently. We know plastic production is continuing to increase. Plastics are not only pollutants, but also amazing, right? They have incredible properties that we use for a variety of reasons, including to help combat climate change in some ways, right? Because these are lighter products to ship. And some single-use plastics, I think we would all agree with, right? Like if you're at the doctor's office and they're gonna use plastic for you once and throw it away, I think we're fine with that. But I think recently, among the reasons folks are more interested in it, has been a little harder to ignore the fact that we don't have a circular economy for plastics. The idea that like when you use plastic, it would go somewhere and be repurposed for something, whether that's energy or, it becomes another plastic bottle. One issue maybe folks have heard about in the news is that a good portion of our recyclables in the US were being shipped overseas, and some of those countries said they don't want them anymore. Meaning that we need to actually deal with them. So I think that's among the reasons why folks have been a bit more interested in this recently. Among the reasons why folks might be concerned about microplastics in the environment or in the water, it's kind of summarized here. The first would be physical damage. Like if you have seen some videos or pictures of wildlife where they're showing plastic in their guts, right? It could be clogging their guts or cutting up their insides, making them more susceptible to maybe infections, things like that. I'm not a toxicologist, but they've also shown these other impacts, like when aquatic organisms are around microplastics, their behavior changes, their offspring have shown toxicity. In general, oxidative stress, let alone if it's compounded by the fact that plastics are really good at having something stick to them. But I think the most compelling evidence might not be from what we always think of as the microplastics from like your consumer products, which a lot of which I think is food grade materials, but something more like what they've shown for coho salmon. I'm not sure if folks have been following this as much on the East Coast, but there's a additive that we add to tire rubber, 6 ppd, and it breaks down to 6 ppdq, and that is what they've linked to die-offs of salmon out west after it rains. It doesn't live very long. It has a very short half-life, right, in the environment. But when you have a rainfall event and a ton of tire rubber and this leaches out and it reacts with what comes out of your tailpipe, it's been shown unequivocally to be toxic to fish. So if you were like, am I concerned about microplastics? I'm like, polyethylene, polypropylene, I guess, right? There's toxicologists showing that's an issue, but I would completely agree that there's certain plastics that are not food grade that we haven't tested for consumption that are out there in our water that I'm a bit concerned about. I am an environmental engineer, and among the things that environmental engineers do is design and treat wastewater and drinking water. So another reason folks might care about plastics right now more than before is the state of California passed a bill saying that we need to be monitoring for microplastics in the source water where the drinking water comes from. So that is a big pressure, because if they find it in the source water, the next thing they're going to do is go and look to see if it's still in the treated water. And if it still is, it's not being removed well, along with a growing body of toxicity data. That's what leads to regulations. I don't think I have it here today. Oh, this is the timeline, and they're incredibly behind because something happened in the middle of this, being COVID. But right now there's 4 certified labs in the state of California around the country that can take these samples, and they're starting to monitor their source water in California for microplastics. New York and New Jersey have also dabbled with this a little bit. And I guess one other thing I wanted to say is for the Safe Drinking Water Act, which is what regulates your drinking water quality if you're on public, city water, you don't have a private well at home. Every few years we have the UCMR, the Unregulated Contaminant Monitoring Rule, and public comment and the government has been, microplastics is among the list of contaminants they're talking about including in that, which again is sort of getting closer to maybe regulating this as a contaminant in your drinking water, just like we regulate a ton of other contaminants. So what are microplastics? The definition's been sort of evolving, but folks are falling around here where it's some sort of man-made polymer as opposed to natural polymers, right? Like cellulose is a natural polymer. We don't want to include that. Usually it has an upper size range of about 5 millimeters. Sometimes we'll say there's a lower size range too. And then if folks are talking about fibers, they've decided those could be longer because of their really that aspect ratio, that small dimension could make them get places. Solid, insoluble in water. There's a lot of polymers that are soluble in water. Actually, we use a ton for drinking water and wastewater treatment. So that makes those utility folks okay. And then the EU has definitions about biodegradable, and folks are saying maybe we shouldn't include those. And industry is also certainly lobbying to have other things excluded from this. In terms of measuring this, most of the data available is from about 300 microns to 5,000, mostly because a lot of this, that work I mentioned in the 1970s, they used 330 micron nets to do zooplankton sampling. So a lot of folks use that as the first method. But California, with their monitoring, is saying we approve of methods down to 20 microns. How does that relate to health? Well, when you get smaller, maybe that's where particles could do not just wouldn't be just you eat them and they're in your gut and they pass out with your feces, right? It'd be where you could have translocation into other tissues, right? Which would obviously have other potential systems effects on someone. Again, engineer, maybe you don't want to hear me talk about toxicity, but if it passes out of the gut, it's a different problem than if you're just eating it and it's passing out with your urine or feces. So that happens. Maybe median particle size, folks have reported around that 80 micron size. If we think about the abundance of these particles. As you go down in particle size, you get more of these particles. The conceptual model is that if you have like a large piece of plastic and it maybe gets embrittled, it would break down into smaller pieces of plastic, not necessarily be like degraded into CO2 or something like that. So this is a larger conceptual model folks talk about when they think about plastic pollution, that a good portion of it is coming from land-based sources. That's us, right? What you flush down the toilet, that goes to the wastewater treatment plant that's not removed, what ends up in the storm drains, a lot of which don't have treatment, right? What blows away. A small portion, especially on the smaller particle size range, we know comes from atmospheric deposition too. That's why we think we're finding these tiny plastics everywhere, because they're small. Like when we get down to the smallest size, it's like, it's dust, right? It's that and smaller. Certainly you could have some pollution at sea where there's some interesting stories. I guess I should have included the slide, like when container ships sink, they'll be able to track where the pollution went. And there's a kind of a famous one of a ship that went down in like the 80s or 90s that was carrying Garfield phones. And there's a story like, why are Garfield phones still watching up on this beach in France or something? So look that one up. And then in terms of the fate of the plastics, a thought is that, you know, some plastics we know float, some naturally sink, like PVC or your tire rubber. They're actually denser than water, so they're going to naturally sink. But even for the floating plastics, we think that they ultimately end up in the bed sediments because for a few reasons. One, you could have natural coagulation flocculation, like particles sticking together, getting big and fat and heavy enough that they settle out. That's common drinking water, wastewater treatment technology. It happens naturally in the environment too. Another would be that when biota eat these particles, if they're passing out with their feces, their fecal pellets, those sink too. So if they're incorporated into these pellets that are already sinking, then the thought is that the net export would be down. And then the one other process that's mentioned here that maybe will come back later would be UV. I think y'all have probably all dealt with some sort of plastic that was maybe old or sitting outside, and when you picked it up, it crumbled, right? Some plastics have more UV stabilizers and additives to them to prevent that. But even with those, eventually a lot of plastics will become embrittled and break into smaller pieces. So I was going to tell you two stories today of how my lab got to be doing this. And the first one's what I would usually tell people at like scientific conferences, which is this. I had a good friend from grad school who was working at a drinking water utility. Folks were calling and asking, are plastics in our water? I thought it was all due to personal care products at the time. And I said, I've never seen a microbead in the water, but it's very hard to get access to like people's private drinking water or the treatment plants. And being a new faculty, I was like, let's just go out to the river because that's the source of a lot of drinking water. To find out what we were looking for, we went to the store and bought all those personal care products and extracted the particles to see what they looked like, right? We knew that microbeads were being added to these products. That first, I was like, oh, they're not all beads. There are also a lot of fragments in some of the ones we looked at, and sent students out to the river to go sampling for these. And at that point, we used mostly visual identification. And from there, we built some collaborations and got to do this on a bigger level and better. So part one for today is estuary case studies that we've done looking for plastics in waters around New Jersey. And the second part would be like another version of the story of how we got here. Like how did me as an engineer actually end up working on plastics? What background led to that? So for the estuary case study, we have a team. It's me, a chemist at Rutgers Camden, Georgia Arbuckle, Grace Saba, who's a marine ecologist, and then Bob Chant, who's an estuarian physicist. And when I went and asked Bob, like, hey, do you want to go look for plastics in the environment? He says, yeah, you know where we should go? We should go to river plumes. So we have fresh river water coming out to the coastal ocean. You get these frontal zones where particles tend to accumulate. And they know this from years of doing sediment transport. So he's like, you know, it's very likely that this is exactly where plastics accumulate as well, because it's where sediment particles accumulate. And you can always make the argument that this would also be a good place to be looking because of those particles and nutrients coming all together. That also means that it's an area of high biological activity. So you could say, oh, if we want to see where this could be like entering the food web, right? We'd say, let's go and sample freshwater. Let's ride it back and forth across these frontal zones and go out to the coastal ocean and see, like, can we find plastics? What are the concentrations? Do they follow the pattern we'd expect? And does it also biologically follow that pattern where, like, if we sample biota from there, that they'd be more likely to be carrying more plastics in their guts? So benefit of working with the marine and coastal folks is they get us out on the research vessel for Rutgers, the RV Rutgers, and we'll go out and they'll do fancier measurements too, but I wanted to point it out, like, this is what some of those frontal zones would look like, kind of like scummy areas, or where here, this is sort of like seagrass. It's where debris gathers. So maybe if you're ever out on a boat, you've noticed these before. And then Bob would do nice measurements to also prove that there's a really sharp salinity gradient that goes with these as well. And when we're out there, usually sampling with nets for this, this is where our nets break because there's so much debris. This is also where, if we're ever finding like a balloon or a water bottle that clogs our nets, this is where we catch them usually. And we'll collect water samples from there and other areas, but the water samples from here, like, even visually in the field, we're like, this does not look like clean water. You can visually see the particles there. So I'll show you a little bit about a study we did on the Delaware Bay. So this is like Cape May, New Jersey, down south. This is the coast of Delaware River coming down here. And we'd go and sample from the freshwater end of the river across the frontal zone out into the coastal ocean, and the same for here. So from freshwater across that salinity gradient and out into the coastal ocean. And it varies based on flow and other tides and other things. And my lab went out and did the water sampling, and then Grace's lab did the zooplankton sampling. So maybe after your pizza, we want to wake up a little. I told you that sometimes in these samples, we could see the plastics. So how good are you at visually identifying the plastics that we see? So here's 5 particles. These are actually from the Raritan River, which runs through New Brunswick. How many of these do you think are plastics? Feel free to like chat with the neighbor and think about what properties also made you think they were plastics or not. And I'll take a sip of my tea while you discuss amongst yourselves. Okay, you got it. You're locked in. So for some folks, when they do these plastic studies, and our earliest study was with visual ID, and then we saved some of those particles and went back and did it with the spectroscopy techniques we use now. So for these particles, I showed you, I guess 4 out of 5 were plastics. That one I crossed out happened to be like a wool cotton blend when we looked at it. So some of these you were probably like, yeah, I don't think that's a color I would naturally see in the environment, let alone the fact I didn't mention that we extracted these as well. So we should get rid of like easy to break down things. So some of them you could absolutely pick out based off of their properties, maybe the color or how shiny they are or whatever. And some of them it's, I don't know, right? I guess this is level 2. So Price is Right version. What percentage of the particles here that we captured out on the Raritan Hudson do you think are plastics? Yeah, like 100% of the particles I'm showing you there, 10%. You can whisper it to a friend too. Price is Right rules, so closest without going over. So some of these are probably like no-brainers, right? Like that bright blue and the bright green is totally there. And the other ones, I don't know, right? It really would be good to have another technique. So here, 61, did anyone? Did we have a winner? Oh. So that's how we started out. And actually, a fair amount of the studies you've seen in the news, especially earlier ones, were only using visual ID too. Now that we partnered with folks that helped teach us the techniques that weren't in my toolbox to do this. This is sort of what the plastics look like, examples from a study we did with stormwater. So there you can see it's like pretty underwhelming, right? It's not that bright blue and bright green. It's a lot of dull particles that you probably wouldn't be able to tell with your eyeball. So we use vibrational spectroscopy techniques to analyze these particles. This is a chemistry student, so I collaborate with environmental scientists and chemists and the other folks I mentioned. So vibrational spectroscopy, Raman or FTIR, if you take organic chemistry, maybe you'll use some FTIR. It's based off of the stretching and bending vibrations of the bonds in the molecule. There's other ways folks do this as well. Mass spectroscopy is another one, but we're going off of this. And the vibrational spectroscopy techniques are actually as well what the state of California has approved right now for people doing this analysis in water. So for the image I showed you before, I circled a couple of particles and I'm showing you the spectra. So here's polypropylene. So here's the CH stretch for that. Based off of that chemical structure, you'd expect that the bonding for the CH here looks different than the bonding for the CH in polyethylene. And students in our lab sometimes come in with their orgo and sometimes not, but by the end, if I showed them these, they'd know them off the top of their head. And I'm showing you very easy to interpret spectra. It gets a little more complicated than this. So we also use library databases for them. And when it gets the trickiest, we call in Dr. Arbuckle, who is an excellent analytical chemist too. Okay, so we go out, sample the water, do extractions, and analyze the particles like I showed you, and bring it back to what we saw. So here, October 2019, everywhere we went, bubble plots here. So the size of the particle tells you about the size of the bubble tells you about the concentration. So that sampling event, no real spatial variation. We went back out after the pandemic, very pandemic impacted science here. The freshwater, the concentration was still low. Out to the coastal ocean it was, but Bob's guess here is right. We see the highest concentrations at these frontal zones right across that salinity gradient. And this was an environmental science master's students project. To show those data a different way here, microplastics per cubic meter. Everywhere there's a capital P is where we had also found macroplastics, like a balloon or a lighter or a candy wrapper, I think were the ones we saw that day. Where there's stars, that means it broke our net. So we can only estimate, like we can qualitatively describe what we saw. And in terms of the polymer types we saw, it's a lot of those plastics that I'm personally a little agnostic about how concerned I am, right? It's a lot of polyethylene, that's the PE in green here, polypropylene in purple, polystyrene. So those are all the, your 1,2s, recyclable plastics in there. PA is polyamide, so like nylon. PDMS is an engineering polymer, and PVC is, I think you're familiar with it. It's used in pipes and other materials. So that's also the benefit of using these vibrational spectroscopy techniques is we're not just saying like, we think this is a plastic, but like this is the type of plastic, which brings up the question of like, well, what's the source of that plastic? And then I can't answer that question. But I think we'll chat about that in a minute. Students are also folks are getting fancier and fancier with this. Here's also us showing like histograms of the particle size distribution. So at part 9, like where we had the most plastics, the average particle size was also greater than when we went out to like the coastal ocean. And then for the folks that love their chemistry here as well, when we collect the spectra, some of those spectra look like as clean as that one for polyethylene I showed you before, where it's just our CH stretch and bend. And then some of them are also showing extra bands that are indicative of like surface oxidation. So here, this is like a hydroxyl band for like an OH group being added. And that's absolutely what happens with UV degradation. So we're kind of like, oh, can we use these surface signatures to say like maybe how long this had been out exposed to UV? I can't say it was all in the environment, but maybe if you have like a I don't know, a plastic pot that was out on your like front steps, that would be subjected to UV and not pollution at that point, right? So when we do this we can do this in the lab, kind of baking plastics in our easy bake oven of a xenon test chamber that with simulated sunlight. And over time, you see these bond indices grow. It's really just them saying the height of this band to some sort of reference band for the hydroxyl or for a carbonyl. We can use that as sort of like a measurement, a guess. This of 90 days would be like 90 days in the full sun at noon. So it's not like really exactly to time. We'd have to go back calculate and be like, well, how often was that sort of the conditions here? But after 90 days of that like full sunlight, we got up to a bond index of about 0.5, and I'm showing you that here for polypropylene that we also took from stormwater. So we see this wide range of like weathering on the surfaces of these particles, which we're kind of interested in for that. Maybe how long has this particle been out there? But it matters for other things as well. The UV, that surface change usually results in a surface texture change too, which changes how things stick to there, maybe how microbes want to grow on there. And it also would lead to embrittlement, right? So when a lot of the times when we analyze these particles, it's actually, we use a lot of ATR, which is you like get your particle in physical contact with IRE, like the measurement. I guess we're using a diamond for it. And to get it in good contact, quite often we crush it as well, because these are pretty easy to break particles. So we can do the same, sort of taking some of those samples from the Delaware Bay and saying like, hey, if we're in the freshwater or out in the coastal ocean, how old do those plastics look based off of these, like that surface chemistry? And here you can see, I just renumbered the sites from before, but from site 1 to 5, right, you see this increase in the bond index. So maybe still a story of like, hey, these plastics out here are older, right? Because it could have also been plastics just coming in from ocean activities too. Okay. So maybe I'll tell you a little story about the zooplankton we've sampled as well. So for the zooplankton, we work with Grace Sava's lab, and their students do the incredibly tedious work of picking through the zooplankton that they find to count like 50 to 100 of a given species that they identify. So this is like a 250 micron scale bar. And this is what the zooplankton look like on that scale. And then we'll digest them and do some spectroscopy. Except for this, the particles are really tiny, so we don't use FTIR, we use Raman. But you get back something similar like these spectra where each of these bands corresponds to different functional groups. And this is what some of those particles looked like after they were extracted from the guts. And this is Carly, the master's student in chemistry who did all this hard work. And then this is the polymer types that we saw there. So again, a lot of polyethylene and polypropylene that the zooplankton were eating. Is this killing them? We think that, I can't tell you from how we extracted where these particles were. We're assuming that the particle size we saw is probably what they're eating and it's in their gut and it's fast passing out with their fecal pellets, not like causing other systems issues. But it's hard to say. And when we do this, we could also estimate, right? Like, okay, they counted 100 A tonsa. How many microplastics did we find in those extracts to try to estimate like the burden, like how many plastics were in the guts of the individual organisms? Although we don't really know if it was like one really hungry hippo that ate it all and no one else had plastic in their gut. But here, this is instead the the Raritan Hudson. So this is like Staten Island. This is the Raritan coming out, and the bubbles here are for how much plastic we found in the guts of the zooplankton that they extracted. So it was pretty similar across the sites. This didn't really follow our expected pattern of it was mostly in the frontal zones. But this is incredibly laborious. So maybe if we had many more samples, maybe we would have a different story there. But at the very least, we showed that real zooplankton out in the environment are eating these particles. Right? Does it matter? I don't know. At the very least, you could imagine that at some concentration, maybe they're wasting energy eating these particles that are not giving them like a nutritional benefit. I guess if you're concerned about absorbed contaminants that could desorb, maybe it matters. Or if you care about like what I'm more ready to speak about, like the fate and transport part. It's a way that floating plastic particles could end up sinking, right? If they're eating these, passing out in the gut and sinking with the fecal pellets, tells you something about that. Which we thought was pretty cool because most of the reports before this were like folks doing forced feeding studies in the lab, which are very interesting, just not my cup of tea. But this is sort of like the images they would get back. They would feed them like fluorescent, like dyed microplastics that were fluorescent, and they could show you them passing through their gut and passing out. So a question would be like, well, from the polymer types we're seeing, what was the source? And I could say like, well, it's an incredibly widespread range of uses for each polymer type. I can't really say. None of what we're finding is like, this is only used in this industry. So there's really too much overlap, but we will try to do things like say like, well, we're seeing this mix of polymer types, and does that look like what we see in stormwater or what we see in wastewater? So for the Hudson River, or well, Hudson Plume, Raritan River study, this is concentration we saw in surface water, wastewater influent, effluent, and stormwater, and all the polymer types. And then we'll try to do some like fancy stats to see like if we take those like whole stacked bar charts of the polymer types and break it down into 2 dimensions, ones that are like physically closer to each other on that plot, you'd be like, oh, well, that range of polymers looks like the range of polymers we saw in the wastewater effluent or stormwater or whatever. So everything in blue here is a surface water sample, and then the other colors are for different sources. So some of the surface water looked like wastewater effluent, some looked like the polymer range we saw in influent, and some looked like the stormwater. So just some ideas of how we're thinking about this. Maybe based on time, I'll skip a few of these slides I had on wastewater and stormwater. But basically wastewater, or breeze through them, wastewater treatment plants are not designed to remove these particles, but are doing a pretty good job anyway. This is from a literature review. I felt like wastewater treatment plants were getting a lot of blame for this, so we decided to go out and look a bit more at stormwater. Just thinking, looking around, right? There's a lot of people aren't picking up their plastics, right? And that's a lot of stormwater is very lightly to completely untreated. So students in my lab will go out and do things like find stormwater outfalls, sample across the storms, do extractions and analysis. And the story here is pretty similar to what we said before. As you go down in particle size, the particle counts go up. And then they'll also, being engineers, try to think of like, well, how does this relate to how long it's been since it rained, or how deep the rainfall event was, things like that. In the stormwater, we had an excellent chemistry student who was able to go down smaller in particle size and found an incredibly wide range, in my opinion, of polymer types in there, including ABS, which is like tire rubber particles, which is the one I'm like, oof, well, no surprise it's in stormwater, but like maybe not something we want just going out into surface water completely untreated. I know we don't have coho salmon here, but there's a really wide range of plasticizers and additives in these particles. Maybe it'd be good if we weren't just letting them all run off. So some conclusions from this work. Yes, my friend asked, can we find microplastics in drinking water? I'm not sharing with you data from our work trying to do that, but we can absolutely find it in surface water. Say something about the sources, but really the pathway of entry, because we're the sources, right, of the plastic. And say something about the polymer types and the uptake into biota. So this is several students worked to get the data here. Almost all of them were master's students and some undergrads we hire for the lab as well. My collaborators, and then thanking everyone that pays taxes, because this was almost all funded by, federal and state research dollars. A little bit by the Water Research Foundation, which drinking water and wastewater utilities pay into to get research done that they need. So then for part 2, knowing that this is part of the WISE program, right, I was going to share